Hello, everyone, and thank you for joining us today for another A Spotlight presentation. My name is Rouzbeh Afrasiabi, and I will be your host. Today, we have Jesse Vick, who is research scientist at Salesforce, and he will be talking to us about his recent research. With that brief introduction, and without further ado, I hand things over to Jesse. Jesse, please take it away.
Thank you. So today I'll be presenting BERTology Meets Biology, Interpreting Attention in Protein Language Models. This is joint work with Ali Madani, Lav Varshney, Saimeng Zheng, Richard Zocher, and Nazneen Rajani. This paper draws from two very different research areas, BERTology, which comes from natural language processing, and biology. In the first half of this talk, I'll give intuition and background about each of these two areas, and the second half, discuss how BERTology can inform biology. So I'll begin by giving some background on BERTology. But to understand BERTology, we first have to talk about BERT. And so BERT is perhaps the most well-known of modern natural language processing models. It's designed primarily for text classification problems. It takes as input a sequence of words or word pieces, and it gives some prediction, in this example, whether the input is spam or not. So BERT belongs to a class of models that's known as transformers. I won't really go into details on the architecture, but just mention that the defining characteristic of these models is this self-attention mechanism. And I'll give some intuition about that a little bit later. And there are also many other transformer models besides BERT. So most famously, GPT-3 has been in the news as a very powerful text generator. One of the challenges of transformer models and neural networks in general is that they're very data hungry. They require a large number of labeled training examples, assuming you train it from scratch. And of course, labeling data is very expensive. But like in most domains, there's a large amount of unlabeled data available. If you think about all the text that's on the internet, for example, the question is, can we learn something from this data, from this unlabeled text? And so the most common approach to do this unsupervised learning is through language modeling. In its most basic form, language modeling is simply the task of predicting the next word in a sentence. And this language model can be trained using the actual next word in the data as the training label. So this is self-supervised learning. So why might this be useful? Well, in order for the model to guess a reasonable next word, such as rolled in this example, it needs a significant amount of semantic and syntactic knowledge. For example, it would need to understand the concept of co-reference, that it refers back to ball, for example. It would need to understand that kicked indicates this event occurred in the past. And it would even need some world knowledge to know that a kicked ball rolls. All the information it learns about syntax and semantics would not just be useful for a language model, but really for any task in NLP. So the whole goal of pre-training is really to be able to gain this knowledge through language modeling and then transfer it later to downstream tasks that are more practical. We can hypothesize about what the model is actually learning, but we don't actually know unless we look at the model. Unfortunately, transformer models are quite opaque. Due to their size and complexity, they're effectively black boxes. Because of this challenge, there's been a lot of work focused on trying to interpret these transformer models, trying to look inside the black box. And this is sometimes referred to as BERTology, literally meaning the study of BERT. But in this talk, we'll mean it to refer to the study or interpretation of really any transformer model. So one approach to interpreting transformer models is through their attention mechanism. I talked about this briefly earlier, but now I'll give some intuition about this. So let's say you're a transformer model and you're trying to finish this sentence. First of all, one of the things you have to figure out is what the word it refers to. What attention enables the model to do is to kind of look back into the context in order to understand the current word it's processing. So in order for the model to understand that it refers to ball, it can actually look back at the word through its attention mechanism or attend to the word. This attention mechanism is associated with a weight on a scale from 0 to 1, which really represents the strength of the attention. In some sense describes the importance of that word in the context to understanding the current word. But this is just one example of the types of linguistic patterns that attention can capture. They've been shown to capture a wide variety of syntactic, semantic, and structural patterns. In some sense, attention gives you a blueprint of sort of how the model understands language based on how it forms relationships between words. Because really what natural language understanding is about composing a larger meaning from individual words, and attention is how the model achieves this. Just a few more technical points about attention that I wanted to mention. Attention in these transformer models is multi-headed. That means there are actually multiple copies or versions of the attention mechanism, which are called heads, and each captures a distinct set of relationships between inputs. So what we're visualizing here is the BERT base model. It has 12 layers and 12 heads for a total of 144 or 12 times 12 distinct attention mechanisms. So as I was saying earlier, we can use attention to interpret the model. And one way to interpret attention is just to look at it. So this is a visualization of attention. In this case, for the GPT-2 model, which is a text generation model. On the left, we see the attention for the sentence we just looked at. The girl kicked the ball into the woods. And the lines and the highlights represent the attention weights from the highlighted word it. On the left side. And darker means more attention and lighter means less or no attention. So if you look on the example on the left, you can see that most of the attention from it is going towards ball. The attention to the first word is actually an artifact of this model, and so you can disregard that attention. But it seems like it's targeting co-reference on the left. And in fact, if you look at the attention on the right from the word she, most of the attention is directed towards girl. So this is interesting. It seems to be capturing co-reference, but why might this actually be useful? So let's look at a different example. On the left, we have a sentence. The doctor asked the nurse the question, he. Besides the first words, most of the attention is directed towards doctor. But on the right, we have a different example, which is the same as the first example, except it ends in the word she. And in this case, we can see that almost all of the attention is directed at the word nurse. This points to the unfortunate fact that many of these language models are rife with gender bias. And in fact, when you look at the output of these models, so these are the same sentences we just looked at, they generate continuations that presume the relationships we just discussed, that she refers to nurse and he refers to doctor. And so these are just some examples, but it shows how these kinds of attention patterns can reveal, in some cases, problems about these models, and hopefully point to solutions to how to improve them. So now I'd like to move into something completely different, which is biology. And so first, some background on biology. So our work is specifically concerned with proteins. So let me talk a little bit about proteins. Proteins are complex molecules that play a critical role in the function and structure of all organisms. Understanding proteins is really important for developing disease therapies and also has some other applications. What's important to understand is that there are sort of two fundamental ways of looking at proteins, as shown on the right. On the top, we have the actual three-dimensional structure of a protein sequence. So these are actual physical entities that exist in the real world. They embody a three-dimensional structure. But if you sort of uncoil that structure, as shown on the bottom, all of these protein sequences can be represented as a sequence of these distinct amino acids, of which there are 20 distinct varieties. And so you can actually model protein sequences just like you model languages. Whereas in language, the individual components are words. In protein sequences, the individual components in the sequence are amino acids. And so we can actually use the same methods from NLP and apply those to protein sequences. For example, language modeling. But instead of predicting the next word, we actually predict the next amino acid in the sequence. And this way we can do the same thing that we did for NLP, which is leverage the large amount of unlabeled data. There's actually a few billion unlabeled protein sequences where only information available is the raw amino acids. So by doing this language model, we can form these representations that are useful for other downstream protein sequence modeling tasks. But the question we're trying to answer is, what is the model actually learning when it does this language modeling? In some sense, to draw a parallel to NLP, what are the syntax and semantics of proteins that this model might pick up? To do this, we can use techniques from BERTology that we just discussed, in particular analyzing attention in these models. So we conducted an experiment where we used a BERT-based protein language model obtained from the tape repository. This was pre-trained on 31 million unlabeled protein sequences using what's called masked language modeling. Simply put, masked language modeling predicts symbols in the middle of the sequence versus traditional language modeling that predicts the next symbol. And this is what BERT does, because it's a bidirectional model, which I won't go into detail, but that's the reason behind the masked language modeling. And so what we did was we constructed a data set of 5,000 protein sequences, and we analyzed the attention induced by these sequences. So we started by just simply visualizing the attention. And so that's what we show here. We have 3 specific attention heads, and the attention that was induced from a particular protein sequence. This is the same visualization we saw for natural language, except that instead of having words, we have these amino acids. And so we're showing the 1-letter codes of each of the amino acids here. And you can see when you look at these attention patterns that some seem to be very interpretable. So on the left for head 6-1, we can see that this attention pattern seems to target the previous amino acid in the sequence. And this is a common structural pattern we also see with natural language models. And then there are other fairly straightforward ones as well, as you can see here. But there are also attention heads where there's clearly a pattern, but it's not exactly obvious what it means. So in this head, we can see that attention spans a relatively long distance in the sequence. And it's very concentrated, but it's not immediately obvious what it's doing. And the reason for not being obvious in this case is that we're looking only at the sequence. We're missing out on the fuller biological context, in particular the 3-dimensional structure. So this 3-dimensional structure is shown here on the right. And we can actually take the attention we visualize on the left and project it in this 3-dimensional space. And what we can see is that attention very clearly targets these spatially close amino acids, which are actually far apart in the sequence. So why would a language model want to learn these sort of long-distance relationships for these spatially close amino acids? Well, it turns out that amino acids that are close to one another interact with one another chemically. And these interactions create dependencies between the types of amino acids that might occupy these particular positions in the sequence. So this provides additional context to the language model to predict these amino acids. And in doing so, the model has essentially reconstructed the 3-dimensional structure of the protein, even though it only knows about these amino acid sequences with no additional information. This is actually a known type of relationship, which is known as a contact map. The formal definition of a contact map is that it consists of amino acid pairs that are within a certain distance of each other spatially, but far apart by a certain distance sequentially as well. So we just looked at one particular protein sequence where attention seemed to be aligned with contacts. But the question is this just one particular example, or is this more of a systematic alignment between attention and these contact relationships? And so to determine this, we conducted a more empirical experiment using the data set we mentioned earlier to see how attention aligned with these contact maps. And here we see the results. So this heat map shows each attention head in the model. This is the 12 layers and 12 heads I mentioned earlier. And within each cell, it shows the proportion of attention that is aligned with contacts over this entire data set. What we can see is that there's one particular attention head, the 12th layer's 4th head, that is very strongly aligned with contacts. And this is exactly the head that we looked at earlier. So it does seem that this is a very systematic alignment that the model learns between these two features. This is another visualization of another protein sequence where we can see this particular head. And we can again see how attention very visually aligns with these contacts, how it connects amino acids that are spatially close to one another. We also found that attention targeted other types of biological properties and relationships. For example, binding sites. So binding sites are portions of protein sequences that bind with other molecules. These are very important because how proteins bind with other molecules largely determines the function of that protein. So the binding sites sort of provide a high-level functional characterization characterization of the protein sequence. And so attention has sort of spontaneously learned to target these binding sites. It's a very rich source of information for the model. Again, this was without any underlying knowledge of these biological properties. So I'd like to make just one final point about how this method can actually potentially aid with scientific discovery. So one of the key differences between biology and NLP is that NLP aims to understand capabilities that humans already have, which is understanding and generating language. In contrast, proteins are an ongoing subject of scientific investigation. So interpreting these models can potentially aid in scientific discovery by identifying previously unknown relationships or providing more nuanced forms of known relationships. For example, the contact maps that we discussed earlier. And these contextual visualizations, like this three-dimensional embedding of the attention, can also make these representations more accessible to domain experts to help with this form of scientific discovery. And we think there's a lot of potential to extend this kind of approach to other disciplines beyond biology as well. And I wanted to conclude just by giving some context for this work. So this is part of a broader effort within Salesforce to do research in protein modeling. And I'd like to just kind of point out a few resources you can look into to learn more about this work. With that, I will thank you for your time, and I'll take any questions.
Thank you so much, Jesse. Thank you for your excellent presentation. So just give me 1 second to check if there are any questions online. Just one comment on YouTube. They say, thank you for your presentation. This is from Fred Monroe. He says, thank you for your presentation. Surprised to see Salesforce working on protein sequences. I don't know if you want to provide any information as to why Salesforce is working on proteins and biology, so on and so forth. Thank you so much.
Sure. So Salesforce is interested in basic research in many different disciplines. So this has to do with a variety of reasons, but one of which is that biology has a lot of potential to help with disease therapies. So as their general interest in AI for good, they've sort of identified biology as one area they want to pursue. And they do conduct research in other areas as well that doesn't necessarily directly apply to the Salesforce product, or it doesn't immediately apply but might in the future. So this is just one kind of general area of interest for Salesforce.
All right, thank you so much. So to go back to the research that you have done, one of the things that might be interesting for our audience to understand is the way you're analyzing the attention. I believe Formula 1 in your paper. If you could explain to us how you're calculating the attention for in your experiments.
Sure. So in Formula 1, I define the alignment, I believe, between the attention and particular properties, and essentially it's a weighted average of the alignment based on the attention values. And so let me just bring this up myself so I can review it.
Thank you so much.
Yeah, so it's essentially saying that for any arbitrary property, this can be a pairwise property like contact maps, or it can be a property of a particular amino acid like binding sites. This is an equation to compute the alignment between that property and the attention. And essentially, it's simply a weighted average of the proportion of attention that is aligned with that property versus the overall amount of attention. And so that kind of gives you a sense of the alignment between attention and that particular property, which is what we're trying to analyze with this experiment.
Okay, thank you so much. So just for my own better understanding, would it be possible to include that information as like information about specific amino acids, let's say hydrogen bonding, hydrophobicity, that type of information as part of the data that is being processed? I don't know if this requires training the model again or not, but could you explain if that would be possible?
Definitely. So this is sort of a higher level initial analysis into sort of the most fundamental biological properties, but there's a lot more nuances to all of these properties, and those could certainly be areas to further explore. They wouldn't necessarily mean training another model, so I think it would be possible to pretty easily extend this work to do that kind of analysis.
Excellent, excellent. Another question I had was in regards to the way you use the model and whether or not you made any modifications to the architecture. Because I noticed that it's similar to the base BERT model. It has 12 layers, I believe, and 8 heads. Is that correct?
It has 12 layers and 12 heads.
Okay. So no modification to the base BERT model, correct?
Right.
Okay.
So the pre-trained model from tape that was provided to us, and they did not change the architecture from the standard NLP model to my understanding.
And do you have any plans in the future to maybe modify the architecture to see if maybe that you would gain better insight or get results that are more accurate?
Yeah, I think so. So as I just said, the model really hasn't changed at all between the original NLP model and the biology model. And you can imagine that language is very different from protein sequences in some ways. So I think there is a lot of potential to adapt these to the particular characteristics of biology. And that is something we're working on currently, but it's not something we've shared yet. But that is a very valid point. And I think that's one really promising direction, is to kind of make these more appropriate for the biological context.
Excellent, excellent. And would it be possible for you to go to one of your slides where you show the percentage of each head's attention for 2 of the amino acids? I think it's proline and phenylalanine. I'm not sure if it's part of your slides or not. Hopefully you have it as part of your supporting slides. Yes. So let's say in case of amino acid proline, would it be possible for you to just give an overview of what the graph means and how it is related or relates to the later results that you have for similarity and how you compared it to Blossom 62?
Sure. So if you look at the top figures, these are the alignment. This is the alignment between attention and particular amino acids. So this is the proportion of attention that's directed to these particular amino acids in particular attention heads. So on the upper left-hand corner, you can see the attention map for proline, or the attention alignment for proline. And on the right, you can see for another amino acid. And so what these show is that there are particular attention heads that tend to target particular amino acids. They're very specialized in these amino acids. This is sort of analogous to an NLP if you had particular attention heads that targeted particular words or particular types of words. And so one question we had was, does this reflect memorization of the model of particular amino acids, or is it sort of learning more fundamental properties that might be shared across different amino acids, which would suggest that the attention patterns between similar types of amino acids, which have similar properties, would also be similar to one another. And so that sort of spurred us to do an analysis where we looked at the pairwise correlations of the attention patterns between different amino acids. So in this example, on the top, we have these 2 particular amino acids. You can actually compute the similarity of their attention patterns by just taking the Pearson correlation of the 2 heat maps, essentially. And that gives you a sense of how similar the attention patterns are. If you repeat that process for all pairs of amino acids, we have what we have. We're showing the lower left corner, which is this attention similarity heat map. And what that shows is for each pair of amino acids, so the amino acids, again, are abbreviated by these 1-letter codes. Each cell in the heat map represents that correlation of their attention patterns. And so it sort of represents the similarity of attention. And on the right-hand side, the lower right, for comparison, we have what's called the Blossom substitution matrix. And this gives some intuition about sort of how similar amino acids are to one another structurally and functionally. This is an existing measure in biology from, I think, many decades ago. And it's constructed by composing multiple sequence alignments and identifying the amino acids that sort of behave or are aligned similarly. But it gives you a sense of the similarity between amino acids. So what we did was we compared our computed attention similarity on the left to the BLOSSOM similarity scores on the right, and found that those had a high degree of correlation. And so this suggests that the attention mechanism is capturing these sort of fundamental similarities between amino acids in the same way that's captured in this known BLOSSOM matrix.
Excellent, excellent. So we have some questions online. First one from Shahzad Hosseini. She's asking if you aim to use the results from this work for maybe in the field of drug discovery, or would you be able to explain how these results could be used in the field of drug discovery?
Right. So I have to say I'm not a biologist, so this is just my own particular thinking about this. But I think the focus of this work is really just on forming a more basic understanding of how the models work. It can also point to, as I mentioned with scientific discovery, pointed to kind of new and sort of as yet unknown relationships in these protein sequences. And so that can give some additional insight that can give a kind of better overall sense of proteins for doing drug design. And as I also mentioned, some of these attention heads are aligning with known properties like the contact maps, but they are doing it in a more sort of nuanced way. So their initial definition, the actual definition of contact maps, is very much a rule-based system. If the distance spatially is less than x and the distance sequentially is greater than y, then it's defined as a contact. Whereas we can have this more sort of nuanced definition of context. So giving this additional kind of subtlety to the measure could potentially add, again, more context to doing the kind of analysis that's used in drug discovery.
Thank you so much. So the next question is, they say that their understanding is that the focus of this is on, or the work is on things like binding sites, but how different does it consider binding sites with some small mutations like ones with which prevent a virus from binding? A bit convoluted, but let's see if...
So yeah, again, my knowledge of biology is somewhat limited, but I will say that we've done some further analysis on post-translational modifications. I'm not sure if this is what the question asker is getting at, but we do find some alignment to these post-translational modifications as well. So I'm not sure if that exactly answered your question.
Yeah, I'm not sure either. Let's take a look at the next question. It's from Abhilash Nandi. I hope I didn't butcher his name. They ask, I had a doubt regarding pre-training. How do you tackle the problem of catastrophic forgetting when you switch from a pre-trained LM trainer on general Wikipedia to pre-trained on clinical domain? I think the question, I think they believe that you have used the base BERT that was trained on just Wikipedia, which is incorrect. It's actually a version of BERT that was trained on proteins. Is that correct?
Right.
And the second part of their question is talking about catastrophic forgetting. I don't know how that, because the original pre-trained model was trained on protein information. I don't know how much catastrophic forgetting would affect the results. At some point, it will suffer from it, but I will let Jesse discuss that. Jesse?
Right. So we didn't really look a lot at the fine-tuning mechanism itself, but That is a general challenge with fine tuning is potentially catastrophic forgetting. And I think there are some methods, sort of like layer-wise unfreezing, to kind of reduce that effect. And I think those could potentially be applied to protein sequences models as well. But I'm just not really that familiar with that particular work.
Yeah, and specifically for this work, you haven't done any fine tuning based on what I understand, right?
Right.
So you were not concerned with catastrophic forgetting. It's not something that they had to deal with. Neil asked, I recently worked on a genetic attribution related problem where I thought of working with a BERT related model. However, the data set I felt was not big enough. How big a data set did you work with?
So there's a few data sets that were involved in this pipeline. So one was the original data set of protein sequences that was used to train the language model. That was something that the tape repository took care of. And that was about the size 31 million. So generally for that size of data set, you need something really large when you're training a language model from scratch. For what we did was to analyze attention. This was sort of a post hoc process. So we really just needed a large enough data set to get statistical significance, which we did achieve. And so in our case, the data set of 5,000 protein sequences was enough for that kind of post hoc analysis. But I think for the training from scratch, you generally need really large data sets. And there's now even larger data sets that are available in the billions. But then it becomes just a challenge of having enough compute to train those language models from such a huge data set.
Yeah, just for clarity, I just want to mention again that in this work they haven't done fine tuning of the base BERT model that was trained on proteins. So what did Jesse and their colleagues have done is extract information, and they have done a brilliant work analyzing the results and figuring out the different layers of information you can extract from attention from sequences of proteins. Let me check if there are any other questions. Shahzad Hosseini, she's asking, in any insight how this type of work can translate to materials? I think they're asking if what would be the application of the results that you get from this work and whether or not it can have real applications.
So I think there is some applications of protein modeling to material science. It's not really my area of expertise, so I probably can't shed a lot of light on that particular application.
So just to give a bit of information regarding why this information is useful and how it can be used in drug discovery and materials science, the type of information that has been extracted here can be very valuable. Because you're dealing with what they have here is just sequences of amino acids. This is barely any information. They're using very little information. As you can see from this slide that Jesse is showing, they're extracting information that can be highly valuable. So, and they have just analyzed 5,000 proteins. So the information that you extract from this work can be useful. And the amount of data they have used is not a lot, but the information that they have extracted is highly valuable. This information can definitely be used for drug discovery because of the interactions that they have been able to visualize and figure out. As Jesse mentioned, this is preliminary work, so they can start injecting more information into the model, modifying the model, so on and so forth, to build on the work that they have done here. So let me just check if there are any other questions. There doesn't seem to be any other questions online. what I wanted to discuss was the binding results that you got in regards to attention. I believe this is figure 4 in your manuscript. If you could just shortly talk about the results that you got from binding, and I believe you have done more work on this, but they have just included a bit like portions of the work. So Jesse, if you can just talk about the binding site results, that would be great.
Sure. Are you able to see the slide I changed to on binding sites?
Yes.
Okay, great. So we did a similar analysis to binding sites to what we did for contact maps. And we also found that there were several attention heads that really specialize in binding sites. The difference with contact maps was with contact maps, there's one specific attention head that really targeted that. Whereas with binding sites, it was very broadly spread over mainly the deeper layers of the model, where a large percentage of attention was focused on these binding sites, which is really disproportionate to their occurrence in the data set. So I think something like 4 or 5% of amino acids occur at binding sites, but in many cases, up to 20% of the attention was directed to these binding sites. And so we did find that they seem to be particularly important to the model. And so one hypothesis is that, as I was mentioning before, binding sites describe sort of the higher level functioning of the proteins based on how they bind to other molecules. And so you can imagine that these particular areas would be very conserved over time as these proteins evolved in order to preserve the original functioning of the protein. So as highly conserved regions, these provide very kind of noise-free information about the protein sequence that the attention mechanism can kind of extract for use in language modeling. And so that's why we believe that these binding sites are such an important part of these transformer models.
Excellent. So one question I had in regards to the results you're showing right now and previous results is the layers and heads that end up providing the most amount of information. It seems like as you move on to the higher level layers and final heads, the amount of information decreases. Is that correct?
The amount of information decreases, you said?
Yes. So the majority of the chunk of information that you have is between, is in the layers in between, as in the head, and also heads in between. Is that correct?
Well, so in this example, most of the information is in the top half of the model, and so that would be kind of the deepest layer. Yeah. So layers 7 through 12, and this is the same, even more so for the contact maps, where it's really in the final. And this is generally what you see. I think I have another slide where I kind of show this. So on the left-hand side, we can show how the attention sort of aligns with these different higher level categories. And we can see generally, there's more alignment, especially for binding sites and the deeper layers. And this is generally what we see with transformer models. This is sort of how they work, that they form the deepest, most meaningful representations only in the deepest layers, once they've had enough layers to kind of process and reprocess the information to form those representations. And so generally it's the last layers or the middle layers that have most of the useful information. That's correct.
Yeah, excellent. So in case of binding site and the turn, bend, and helix, there's a drop in the amount of information when you reach layer 12. Do you have any hypothesis as to why this happens?
Well, I think part of it is that they've sort of the attention has sort of switched over to contact. So there's a limited number of heads, and they can only attend they have a certain amount of attention they can sort of expend. And so in the last 2 layers, you can see that they're really focusing mainly on the contacts. And so because of that takes away attention from the binding sites.
Based on what you just explained, my understanding is that the layers and heads become specialized in extracting specific information. Is that correct?
Yes. So that's sort of a general characteristic of transformer models. We definitely see that in NLP as well. And I think it's one of the more interesting parts of transformers because it makes them more interpretable. And also potentially, more controllable if you can manipulate those specific specialized attention mechanisms.
We have another question online from Xiang. He's asking, he says he's curious about training process. Have you tried training with large batch sizes, or do you use a similar setup with that of the original BERTs, which I assume is slower? I think this is, again, here they have not trained the model again. They're using a pre-trained model. They have not done any tuning. So they're focused on analyzing the results they get from attention. Is that correct, Jesse? Do you want to expand on that? I think there's the misconception among our audience as to whether or not you have done training. I think it's best if you explain it again.
Yeah, so this is purely post hoc analysis. We've just taken an existing model that's already been trained on language modeling and shown what the model is already discovering on its own without us actually having to do anything to the model. And that's kind of the interesting part of this, is that there's no biological insights that we provide to the model, no particular architectural changes. It just learns all these properties spontaneously. But because of that, we were able to just use this pre-trained model from the tape repository, which we appreciate that they made that available. And so it makes this kind of type of work more accessible to people in general. You don't have to train your own models. You can just sort of analyze existing models.
Excellent, excellent. Thank you so much, Jesse. And I would like to thank the audience who joined us today. It was a great presentation. Hopefully in the future, we can have you on this platform again. With that, I would like to thank everybody for joining us and wish everybody a very good rest of the day.
Thank you.